Yeah, microdosing I think is really interesting. Originally my primary interest is in large doses because I think that sort of transformational potential of psychedelics is really what's so appealing to me. But at the same time, I've worked in cannabis for a long time, and I know that symptom relief and that even regular applications of perhaps less profound experiences can also be really helpful. So I think knowing that these drugs are active, and we'll just talk about psilocybin, but the microdosing study that we did had people who are using a variety of different substances. Psilocybin was the most common, but also LSD, and then also some sort of a smattering of other psychedelics that people would be familiar with. But we know that psilocybin has a long history of use. We know that it's psychoactive at large doses. So it seems like sort of almost low-hanging fruit to say, well, what does it do at small doses? And then we find this growing groundswell of people who are reporting positive effects of microdosing. And there and we'll talk about the placebo and expectancy effects, if you will, in a moment. But what I've learned, also from medical cannabis, is really about believing patients and believing people. Like these substances are different than the typical pharmaceutical pathway where a drug is invented in a lab and tested first on animals and then placebo tested on humans who wouldn't have access to it otherwise. So sometimes I think it's a shame that we hold some of those standards and some of those approaches, because I just don't think they're effective or optimal for a substance that's widely available and regularly used. So then the question becomes, how do you learn about these substances? And the good news is there are a lot of people who've used them. So you can ask those people what their experiences are. And you can do it in ways that are more or less sophisticated. I like to think that, our surveys go beyond, just rudimentary questions into trying to get at some nuances of how people use and why do they think it's effective and what might be some of the other things that are going on that could account for it. So they're rather sophisticated surveys, I think, but they are surveys. So they're testing into people's subjective experience, and subjective experience can be due to a lot of things. And I think one of the concerns that people have said about microdosing, and I can't disprove it, is that people only think that they're getting better, that they have sort of you'd say a placebo effect, but I don't think placebo effect is accurate, even though it's widely used, because a placebo effect suggests someone's getting a placebo and they think that they're getting an active substance, and so the effect is due to their misconstrual that they're getting an active substance. Whereas in our studies, people know what they're getting. They were not getting a placebo; they were getting what they thought they were getting, which is small doses of psilocybin. They don't know exactly what it is, certainly, because it's hard to test the chemical constituents. We didn't have that kind of information. What we do have, though, that placebo-controlled studies don't have, is we have very large numbers, and the hope is that there's, sort of the law of large numbers allows us to tease out some patterns of response, and we also have a longitudinal design. So in our study, the Microdose.me study, which is, I believe, the largest study to date of microdosing, we've published 2 papers in Nature Scientific Reports. The first one is the baseline, comparing microdosers to non-microdosers. And one thing that was really remarkable about the study to me is that we had a large number of non-microdosing participants who were willing to keep up with the survey. So it wasn't just microdosers. So, that goes some way. Having a comparison group goes some way towards dispelling the placebo effect, not just asking people how they felt. People went through the same procedures and answered the same questionnaires, and probably also want to feel well and report themselves in a positive light, whether they're microdosing or not, that they're going to the trouble of taking part in the study. They're not being compelled to do so. So we compared them at baseline. Then we also compared them a month later. One of the benefits of that longitudinal study, longitudinal design, is that you get some of the effects of the tests that we're doing. You could have a practice effect where people do it repeatedly, and that would lead to an improvement. But the control condition was the non-microdosing control condition. We're repeating the tests as well. So there's procedural controls, even though subjects were not blinded to the effects. But you could still say that people who are microdosing, there's an effect to thinking that you're doing something that's supposed to be miraculous, and there certainly is a lot of hype about microdosing, and people who are doing it are going to substantial effort to do it. So if you're putting that much time and effort into something, you're going to think that it's effective. That doesn't mean it's not effective just because you think it is. I mean, sometimes people, I think, go too far and think, oh, it must, you think it's effective and therefore it's not. I don't know if I buy that, but it's absolutely possible that some of the effects could be an expectancy. They could be a combination, almost surely feeling like you're doing something good that's helping you improves your mood. So, but that, like I say, that doesn't mean that it's not a true effect. So there could be a mixture of a true pharmacologically mediated effect that's improving mood and anxiety. Those are some of the big effects that we saw, and that is interacting with a positive expectancy. I think that's kind of how life works a lot of the time, where you do something that you feel is improving yourself, and then you feel good because you're doing it, and because you feel good about doing it. So if you start exercising, you might have an improved mood because you're exercising, but you also might feel better about yourself because you're taking active measures to improve your health. But that doesn't mean that the benefits are solely due to the expectancy that you have that you're going to improve your mood through exercise. So I think that there's, in science, there's always this tension between control and ecological validity. So the more you control things, the more you take them into the lab, you can say, yes, this person, they're equivalent in so many ways. And we know that the group that got the microdose, responded differently than the group that didn't. And that gives you a lot of control, but it's not very ecologically valid. If you were to go into a lab and have to collect your microdose or do all kinds of things that people have to do to participate in those kind of studies, that's also a manipulation that's going to change your mood. And when we're looking at very small effects, so I think the most of the effects of microdosing are small. They're not negligible, but they're small, and so a lot of things could impact that. So I can't say that it's not a placebo effect. What I can say is that, out of the over 1,000 people in the original study that microdosed, on average, they said their mood was better than those that didn't. And I think that at least it shows that there's nothing that is diminishing mood or that's harmful in mood. And if people find it effective, then I suppose part of your liberty to experiment with yourself and your mood and your sense of well-being would be like, try to do something that, if it makes you feel better, keep doing it. If it doesn't, then maybe don't do it. But if people want to know whether or not what others say, and if they want to find out what 1,000 other people say, and if they want to be able to read that in a condensed form, then they can look at our research. But it's not a placebo-controlled trial. Now, some placebo-controlled trials, I will say, have tried to control for the effective expectancy on microdosing, and they've done that in a couple ways. And that's where some of the criticisms have come from. So in one fairly well-known study, they asked people, do you expect microdosing to be effective? And then they controlled through statistical means that are readily available. They controlled for the effects of expectancy. So they took out whatever variation was due to expectancy. And then they said, look, they look about the same. They found the same thing we did, that the microdosers said they're doing better. But after they took out the effects of expectancy, the two groups looked the same. So that could be interpreted to suggest that it's an expectancy effect. However, it could also be that microdosing works differently for some people than others, just like pretty much every other medicine. So it could, because the people weren't naive in that study, they had microdosed in the past. So when they're saying, I expect microdosing to work, it could be because it works for them, and it had worked for them in the past, and therefore it worked for them again. And the people who didn't expect it to work may have tried it. It may not have worked for them in the past, and so it didn't work for them again. So I don't think that even that kind of trial where they take out the expectancy effects, the expectancies might be valid and past experience might be predicting future experience. So that doesn't negate the potential that microdosing has an effect. There's another study where they got people to blind themselves, and it's kind of a creative study. Yeah, it was a very complex and somewhat ingenious protocol, I'd say. And so I think we need all of these different, I'm not downgrading this research, but I'm just saying it's not, to me, any more convincing than the studies that show an effect, especially when the studies that show an effect have so many folks in them. But with this study, just about everyone guessed what condition they were in. And we see that across psychedelic research, is just about everyone guesses when they're in the active condition or in the placebo condition. There's huge levels of correct guessing. And for anyone who's ever had an experience with a psychedelic, that won't be surprising that you know whether you're in the active. I mean, it's sort of this theater that we do with placebo that's a bit absurd, because everyone is going to guess correctly. So that's good. But is it really a placebo control the way it is if you take a medication for some kind of more physical disorder and don't have a clue, except for maybe some side effects, but nowhere near as obvious as this? So I feel like we're trying to fit, this, I don't know what's the phrase, the square peg in a round hole, or whatever it is. We're trying to make the psychedelics fit this placebo-controlled design paradigm that may not be a good fit. And it was the same thing for cannabis too. You're giving people ersatz cannabis or cannabis with the THC taken out, and then that's a placebo condition, and they're meant to smoke cannabis that doesn't get them high, and that's supposed to be okay. And is this, particularly with high dose psychedelics, if people are being asked to put on the mask and the headphones, and there's this huge expectation that something momentous is going to happen, and they're struggling with a serious illness, and then they're in the placebo condition, that seems like that would be, to me, disheartening, to say the least, if not actually, inducing a negative state. So is that more realistic, really, as a way to test it? I don't think I think there's a compelling argument for placebo-controlled design, but I don't think it's the be-all and end-all. And one thing it does really serve is, well-funded research groups. Largely, those are going to be private or privately funded, that can afford to do these huge designs. And if you say that's the only way that we can get at truth, is by, truth, in order to say anything with scientific validity, you need several $1 million. I understand where people are coming from, but that seems to create a moat around who gets to say what's what. And I'm not sure if that's conducive to the best discourse. And, if we look at the opioid overdose crisis right now and the overprescribing of opioids, those all went through clinical trials. And so they missed something. So perhaps it's not as bulletproof as we might think. Yeah, I think we need to question it. I mean, if there's one thing that I think that's hopeful about this whole movement, is that we're questioning a lot of things about healthcare. And I think less evident might be that we also need to start questioning some of the paradigms for research and seeing, who do they serve? What do they serve? Are they putting patients first? How do we get to the truth here with these substances? And keeping in mind risk benefits. So I think one of the good things is we have a lot of research on the risks of most of these substances. Even, a lot of that comes from the illicit market, but nonetheless, we know that the risks are not too severe. So how much research do we need to do before we start to slowly roll out these medicines in a responsible way and keep a close eye for adverse events that we didn't expect, have a critical eye for the positive outcomes, but not keep waiting for someone to develop a patent or to do the ultimate clinical trial that means that they that we need that kind of a vetting. Who does that serve? These new therapies out, and I think it's good that people are being conservative and giving it a careful eye. So it's a balance. I hope there's energy to keep up with some of the studies, and I saw that there's a new study coming out in Vancouver that's very interesting, and I think that one is placebo controlled. I think you have a better chance of placebo control with microdosing than with large doses for sure, but people still guess. People will still know what condition they're in.
Yeah.
And in addition to positive expectancies, you could have negative expectancies if you find out you're in the placebo.